um, um, exterior bran. There's this alurone layer, which is full of living cells. It's a very thin layer, but it's a very important layer because of those living productive cells. There's the embryo, of course, and there's this endosperm, which is effectively dead, but full of protein and starch that goes to feed, well, it goes to feed the germ, but more importantly for us, it goes to feed our bellies. Starch granules make up the lion's share of the endosperm, so very high concentrations of carbohydrate in grains. The proteins that make up the bulk of the difference are highly varied, but we will focus on two classes of protein which are themselves fairly diverse. First of all, the gliadins, and secondly, the glutenins. Let's call the gliadins. Let's say that the gliadins result in the formation of small range, short range, weak networks, whereas the glutenins, which can be much larger molecules than the gliadins, are responsible for the formation of long range, intermolecular, that is to say, between different molecules, covalent networks. And we're going to see that this is all important in the development of bread texture. Glutens are relatively high in glutamine. That's an amino acid. I refer you again back to lecture 1 to see what those amino acids look like. It's high in nitrogen, as you'll see, relative to the other amino acids, and it's easily metabolized. Those of you who are into your metabolism biochemistry will realize that glutamine sits at a crossroads for nitrogen biosynthesis. So it's an important, it's a smart choice as a nutrient to increase the concentration of glutamine in your food stock. Bran, as we've mentioned already, composed of this pericarp and the alurone layer, protein rich as well, not gluten rich, however. Just another pretty picture. Cartoon on the left, electron micrograph on the right. We see that the seed has been broken open so that we can see inside, and we see the structure of the endosperm, of course, full of cells, right? But all those cells are effectively dead and now are just serving to provide food to the germ. Let's talk about getting from the grain to the food. And these are just some nice pictures about how we grind our grain, from the very simple dig out a hole in a rock, pour your grain in, beat the grain with another rock, and then blow away the bran, and you're left behind with this starchy, floury material. You can also use something like this, metate, where again, beat the grain between two rocks. Here's a slight advancement over that, where we can drive the spinning of two halves of this effectively spherical rock, and pour the grain between those two, and they're crushed very finely and separated very easily from the bran in that case. And you can see in this picture, here's the whole grain being poured into a hole. This is the top layer, and here's the bottom layer here, and the spinning, and you can see the flour being kicked out the side. This is a more modern milling apparatus. It's basically motor driven, but the same thing. Two pieces of stone, and you see the flour flying out. So you can imagine the texture of the product that you get from this sort of a device versus this. But they both do the same thing, effectively. Now, in terms of these more simplistic methods of just stone grinding, bran and endosperm are crushed together. This has some important consequences, as we saw. We retain the bran, we retain a lot of nutrients and protein that we would otherwise have lost. But that changes the texture of the flour drastically. In refining, we remove that bran and endosperm, and we're, again, as in the polishing, what we leave behind is only endosperm with no bran and no germ. But again, right, so we lose fiber, we lose oil, we lose vitamins, we lose some protein, but If what we're looking for is being in control of gluten concentrations, which we'll see is important, then we don't care about that stuff. And what we care about is modulating the amount of gluten and versus starch concentration in the endosperm that we leave behind. There are also a couple of other steps. Improving, so-called improving the flour. Exposure to air oxidizes gluten sulfur groups. So we're jumping ahead a little bit here, realizing already that if we refer back to our egg foams, that disulfide formation between unfolded proteins is really important, can be good, can be bad in terms of stabilizing a foam structure. But if we expose to air, we can promote the formation of intermolecular cross-links. So just keep that in mind. So a way of improving the flour is to promote the formation of these cross-links. We can chemically weaken flour if you don't want to form too strong of a network by the addition of the amino acid cysteine. So we've got to refer back to our first set of notes. Cysteine is the sulfur-containing amino acid that is responsible for the formation of disulfides, those intermolecular crosslinks. So if we add cysteine, then we form disulfides, all right, but not between proteins, but instead between protein sulfhydryls and cysteine. So preventing effectively the formation of those intermolecular crosslinks. That is a weakening of the flour. Not going to form a good gluten network. Finally, we can bleach our flour. And so if we refer back to these first two steps, depending on how we grind that grain, we wind up with more or less bran, and that's going to impact the color of our flour. But typically flour is going to be fairly brownish in any case, unless we bleach it. And there are a variety of ways of bleaching it, either naturally or artificially, to whiten the flour. We talked about varieties of wheat. Let's think about what that means in terms of the varieties of flours. Again, hard flours have a high gluten content. I've mentioned the fact already, and we'll explain it in a few minutes, that high gluten content is important in the formation or the development of bread textures. Soft flours have a low gluten content, more starch. These are going to be more useful for textures like that which we experience in cake. I think we all are familiar with the difference in textures there. Breads tend to be chewy, sometimes firm. Cakes are good, considered to be good if they're nice and crumbly. That has everything to do with the amount of gluten protein that's available. And we can control that by choosing the kind of flour we use. So on the left here, I'm showing you a variety of different kinds of flour. Some of these are obviously derived from a particular kind of wheat grain. Others it's less clear. The protein content is what I want to focus on. Whole wheat flour, 11 to 15% gluten, which is relatively high. Excuse me. The bran that might be left behind in whole wheat flour can affect that gluten formation. However, that's another story. But we see that, the bread flours, these are going to have on the high end of gluten concentration. On the low end, we see pastry and cake flours, 5 to 10%. And then, interestingly, what we all know of, or most of you are probably familiar with, is this all-purpose flour that we see, and it's a compromise. So we cook with all-purpose flour, we make our cookies with all-purpose flour, we make our cakes with all-purpose flour, we make our breads with all-purpose flour. But we're doing ourselves a disservice by taking that middle road instead of selecting the ingredient that's appropriate for the task at hand. Now, most home bakers don't take that into account, but if you're really interested in developing a good bread recipe, I suggest you go find bread flour and cook with that. There are a variety of other flours. Your text goes into these more than I'm going to bother with. Specific to specific purposes and maybe even recipes, intermediate gluten concentrations here. But as your text also describes, you can easily extract the gluten proteins from the overall protein content of the flour. The gluten proteins are not terribly water soluble, so you can wash away water-soluble proteins with water, and then extract the gluten's themselves with organic solvents like ethanol, and that will concentrate the gluten so that you can again manipulate the structure of the food item that you're working on. And we'll see a great example of that in a few minutes. So, what is this gluten? I've been mentioning the formation of these long-range intermolecular networks. Let's think about that. I mentioned that there are gliadins. Again, gliadin is a class of proteins that's very diverse. Here we're looking at 3 different classes of gliadin: alpha, gamma, and omega gliadins. And what we're looking at here is a one-dimensional map of the protein. And for those of you that are interested in a little bit in the molecular biology, we usually consider these to be these boxes telling us something about the domain organization of that molecule. For our purposes, what we really care about is maybe this hashed section representing is almost sort of a spring-like region of the molecule where we form something called a beta helix that can stretch, relax, and that provides some of that sort of toothiness or elasticity to bread doughs. This third domain is marked with these little thumbtacks, if you will. These mark the position of cysteines. So remember, the cysteines are responsible for the formation of those disulfides. So those are the amino acids, the positions at which the amino acid is cysteine. So we see that the gliadins vary in the size of each of these different domains. One thing that I want to mention, though, is that in the gliadins, the disulfide formation is usually considered to be intramolecular. That is, we're only forming these sulfur-sulfur bonds within a particular gliadin molecule, as compared to the glutenins, in which the disulfide formation is intermolecular. That's really important, but let's take another minute to look at the glutenins. First, we see that there's a low molecular weight glutenin, LMW for low molecular weight. In this case, there's also a high molecular weight glutenin, much larger molecule. Not so important for us to think about the relative size of those, but what's more important to remember is that these cysteines that we see cropping up at the beginning and the end of this high molecular weight glutenin can form intermolecular disulfides, that is to say, disulfides between different glutenin molecules. So yes, we have a big long glutenin molecule, but now with the ability to form these disulfides, we can effectively increase the size of that gluten molecule enormously. Oh, this is a repetition, ignore that. Okay, so those are the glutenins. Those are the glutens, and as we'll see, glutenins are considered to be very important for that, for the formation of that nice long-range network. Flours that are high in gliadin relative to glutenin, for example, rye flours, I believe, higher in relatively higher proportion of the gliadins, don't form the same kind of nice gluten structure. So glutens aren't all the same. I guess I should also mention that there are many people who are allergic to the glutens, and there's a variety of disorders. We don't have time to go into that in great detail. Suffice it to say that that's an immune response to particular structures on the gliadins and the glutenins, and can result in, among other things, anaphylactic shock. So those folks are going to avoid food products that have high gluten concentrations. Okay, more on that cross-linking. So I've got a pictorial representation of that. Remember, disulfide formation. So over here on the right we see there's one protein chain, there's a cysteine side chain, there's another protein chain, cysteine side chain, and we can form this intermolecular bond that effectively makes one molecule out of these 2 separate molecules. Well, as I said, the gliadins are smaller molecules. We can maybe think of them as the ball bearings of the gluten network. They're smaller, they form intramolecular disulfides, but the glutenins can form these intermolecular interactions, and that allows us to create this very long-range elastic network. Look, this is a pretty bad slide. I'm sorry for that pixelation. Here's a relatively recent article that describes some other players that might be responsible for glutenin cross-linking. There's some controversy as to how important these tyrosine cross-links are, but I thought it was fun to mention that, especially for those of us that are into our weird biochemistry. This is one protein chain. Remember, this is a tyrosine. I hope you remember a phenyl ring with a hydroxyl group appears in this case to be cross-linked to another protein chain, tyrosine hydroxyl there, right? So there's our cross-link. We can form a variety of types of cross-link. This is another form that's been observed in other cases. I'm not saying that we need to radically revise our ideas of how the gluten structure is formed, but it's not quite as simple as the disulfide links that I think most texts will describe. Okay, a couple of interesting slides. As we're making bread, so we haven't described the process of making bread yet, but maybe it's time to do that. Well, what do? You take flour, you take water, and you take yeast. Those are the three most important ingredients in one's bread recipe. And what happens? Well, you mix all those ingredients and things start rising. Why do they start rising? Well, as we'll see, the yeast is fermenting those carbohydrates into CO2